Good morning, everyone. We are not quite ready. We're still waiting for one presenter, and we'll then start at 9 o'clock Mountain Daylight Time promptly. Jordan, if you could please also have an eye on Victoria coming in. She is our second presenter. I do not yet see her, but we also still have some time. And for all those who are joining us as presenters, as judges, as well as supporters, the part of the audience, if you can please make sure to stay muted and also turn your cameras off. That might be less distracting for our presenters. Here she is. Wonderful. Welcome, Victoria.
Thank you. Sorry, all the doors were locked to my building, so I was struggling to get in.
Yeah,
I don't know.
those things have to happen on those days, right? You are here, you are fine. You still have 2 minutes to open your doors. Please all keep muted. Please, we really don't want to have any background noise for our presenters. Thank you so very much. All right, good morning, everyone, and thank you very much for being here, to our presenters, to our judges, and to the supporters of our presenters in the audience. We really appreciate you being here. Please keep yourself muted and possibly off camera so that we can all focus on our presenters. I'm Inge Wefes, a Senior Associate Dean of the Graduate School at the University of Colorado, Denver, Anschutz Medical Campus. I would have preferred welcoming you in person in beautiful Denver. I hope that this picture can convince you. We have also one of our many sunny days today. maybe another time. With this, I would like to open our 3-minute thesis competitions. We are very often referring to them as 3-minute talks, as at many universities, thesis refer only to the capstone for masters, versus we call dissertations the capstones for doctoral programs. We include everyone here, and we do not disclose in which kind of program our students are enrolled, as we really think it should go by the quality of their presentation, not the kind of program in which they are enrolled. I would like to just share with people a few of our rules. So our students are well familiar with the fact that they have only 3 minutes to talk with 1 slide, no animations. And the rules you can find on the scoring on our websites, and all of our judges have those criteria also listed on a judging page where they will do the electronic judging. I'm most grateful to 4 judges who have committed to be here. Naturally, they are not of the universities where our students come from. Thank you very much to Mark Dallarkaub, who is professor in chemistry and at San Jose University, Jarek Nagel, assistant vice provost for graduate studies at the University of Oregon, Mary Stromberger, vice provost for graduate affairs and dean of the graduate school at Colorado State University, and Hannah Snyder Lynch, director of graduate admissions and enrollment at the University of Oregon. I'm most grateful for people taking time out of their certainly busy schedules to be here. And I'm most grateful to my colleague Jordan Schiefer for moving the slides and overall overseeing the technical aspects of this competition. Judges, please make sure to use the speaker and side-by-side mode, where you have the slide and the speaker in 50% representation each. And audience, please stay muted, and also possibly keep your camera off for now. Our first presenter, we will have 7 presenters, and our presenters come from the University of Utah, the University of New Mexico, CU Denver-Anschutz in Colorado, University of Nevada, Las Vegas, University of Alberta, Chapman University, and University of Idaho. We have a total of 7, and I'm sure we can really look forward to very exciting presentations. With that, I would like to welcome Morgan Nelson, who is a student of microbiology and immunology at the University of Utah. She studies under the mentorship of Dr. Ryan O'Connell and will share with us her research on one big problem and one tiny solution. Morgan, are you ready?
Ready. Have you ever gone on a diet? If you have, you're not alone. In fact, 45 million Americans go on weight loss diets every year, and the diet industry is now a $70 billion enterprise. And yet, so many people fail to lose the weight. What if I told you your genes impact your ability to gain or lose weight? While diet and exercise are still the main treatments for weight loss, we now know that some people have a harder time losing weight than others, making them prone to obesity, type 2 diabetes, heart disease, and cancer. I am studying why this happens at the molecular level. In my thesis research, I have discovered how a tiny molecule called a microRNA has a big impact on obesity. To explain, I want to compare our bodies to a car. In our bodies, proteins are the fuel that keep our cells functioning normally. Messenger RNA is the gas pedal telling our bodies to make more proteins. But we want protein production to stay within the speed limit, and that's where microRNAs come into play. Discovered less than 30 years ago, microRNAs are the smaller cousin to the messenger RNA, and their job is to put the brakes on protein production and stop messenger RNA in its tracks. This is a normal and healthy process that fine-tunes how much protein we produce. But much like the brakes in our car, if microRNAs go out, it can have major consequences. One microRNA in particular may protect us from developing obesity, and we see this in actual patients. People living with obesity and type 2 diabetes tend to have less of a gene known as microRNA 146. If you can think of a better name, please let me know. But in order to understand why certain people have less of this gene, I study mice that lack microRNA 146. And guess what? These mice develop symptoms of obesity and type 2 diabetes despite consuming the same amount of food as normal mice. We are learning that this microRNA controls important functions, including metabolism and inflammation. I'm using this knowledge to test new therapies, including a drug that would replace microRNA 146 to prevent or reverse disease. While the diet culture may not be going anywhere anytime soon, my research is getting us closer to creating a healthier life for people whose genes may be fighting against them. Thank you.
Morgan, thank you very much for this very, very interesting presentation. Judges, please submit your scores, and while you're doing so, remind us, Morgan, in which program you are studying? What was the discipline?
Immunology.
Immunology, yes. And how did you come to work on that topic? What fascinated you with this and kept you going even in times that might have been a little tough, as we have that in science?
Yeah, that's a great question. I was really interested in studying RNA, as I mentioned in my talk. And as I was joining my PhD, or my graduate program, sorry, my professor that I rotated with was in the immunology program, and I kind of fell in love with the immune system. And it was just kind of the perfect marriage of RNA that I wanted to study, plus this whole new field that I was exposed to with immunology and the immune system. And it's really just fueled my passion.
Yeah. And has COVID impacted your work?
Absolutely. I mean, I don't know a person that it hasn't impacted. We were very fortunate to be able to continue a lot of our work through the pandemic. We could stagger our schedules, and we were allowed to still take care of our animals and attend our lab, which made a big difference. But that being said, a lot of what I do is bench work and is in person. And so that was certainly restricted through 2020 and into 2021. So it was definitely impacted. Yeah.
Yeah. So you had to work in shifts to keep the distance.
Yep.
Yeah. Yeah. How much longer do you have to go?
We're shooting for a defense date this fall or in the spring. Hopefully.
Oh, wow. You have our best wishes and good luck with your experiments. I think that will help a lot of people to know what's behind it all. So thank you very much.
Thank you.
Our next presenter is Victoria Penapart. She studies Spanish and Portuguese at the University of New Mexico under the guidance of Dr. Damien Vergara-Wilson. She will talk to us about life or death, an assessment of COVID-19 messaging in New Mexico and potential consequences for the Spanish-speaking population. Victoria, are you ready?
Yes, I'm ready. In March of 2020, we knew 4 things about the pandemic. It was disproportionately affecting minority communities. New Mexico is a minority majority state. 25% of the New Mexico population speaks Spanish. And if we were going to effectively tackle the pandemic, then we need to ensure that all critical health information was being disseminated in a way that all New Mexicans could understand. Because there was such a huge emphasis on access to information, our study analyzed communications from March of 2020 to March 2021. We found that COVID-19 messages sponsored by the government were predominantly expressed in English over Spanish. Our study analyzed translations and broke them down into 4 categories. The first being a complete translation, which means that all information was translated into Spanish and it made sense to a Spanish speaker. Over the course of a year, very rarely did a translation fall into this category. Instead, of the translations that were available, most fell into our next 2 categories, an incomplete translation or an inadequate translation. An incomplete translation is one that translate some stuff into Spanish, but was missing key information that was available in English. So a prime example of this is at our self-swab testing sites here in Albuquerque, where if you're given English instructions, it'll tell you everything from how to swab your nose to how to send the test off to the labs. Meanwhile, the Spanish instructions will only tell you how to send the test to the labs and not how to actually administer the test itself. This brings us to our next category, an inadequate translation, which means there was an attempt to translate something into Spanish, but it just failed. So a prime example of this is when the New Mexico Department of Health utilized a translation software system on their vaccine registration portal. It attempted to translate county names from English into Spanish. So if you were in counties like Bernalillo or Doña Ana, you were relatively unaffected. But if you were in counties like Eddy, which somewhere somehow means windstorm, that gets translated into Remolino. And if you're a Spanish speaker within these communities, it looks like your county is just not available for vaccine registration. And as you can see by this chart, some communities had upwards of 40% of the population being Spanish speaking. This brings us to our last category, no translation whatsoever. In general, most information was just not available in a language other than English, including legal notices and hotlines that were advertised to provide economic assistance during the pandemic, like housing or unemployment. So, what does this mean? Well, the way you choose to deliver a message says a lot about who your intended audience is. We interpret these results to be a prioritization of English speakers and a dismissal of Spanish speakers. In April 2021, our state started targeting populations that weren't getting vaccinated. And due to the lack of foundation of information in Spanish, we weren't surprised to find that Spanish speakers were amongst this list. In a pandemic that's disproportionately affecting minority communities, we need to do better to ensure that this information is making it back to the communities that need it and in a way that they can understand. Thank you.
Thank you very much, Victoria. I think another one of these hot topics, and at least I hope we can let that problem go due to COVID soon, but I think the issues go far beyond that. Remind us, you are in a dual language program?
So I am studying in the Department of Spanish and Portuguese. My specific concentration is Southwest Studies, so Spanish within the Southwest and how that relates to the United States and the socio-political context of that.
Yeah, and how did you come to that topic? What fascinated you?
Well, it ultimately impacted my community. I come from Eddy County, one of the counties that were mislisted on the New Mexico Department of Health, and you just see your peers being affected by this, right? I come from a Spanish-speaking community, and if the people in my community don't have access to healthcare, that's going to affect me and the people around me.
Yeah, and while you are working on COVID-related issues, did COVID itself, not from the disease perspective, but by the interaction restriction, impact you too?
Yeah, of course. I think one of the hardest parts of our study was the fact that a lot of it had to do with going to get tested and going to get vaccinated. And so what was at these sites? And obviously, if you're going to testing sites, we wanted to limit that interaction with people who were sick. So we would usually go if one of us got sick, if one of the people on our team did, but also just community interactions. What was the community telling us? What were they experiencing?
Yeah. How much longer do you have to go until you can graduate?
I graduate in May.
Yay, wonderful. Congratulations. And what are your next steps?
Law school, actually.
Ooh,
so taking this research and fighting the inequalities.
Yes, all right. Well, I think we need people like you. Thank you very much for being here. We are now ready for our next presenter, Adele Price, who is a student of statistics at the University of Colorado, Denver. She studies under the mentorship of Dr. Audrey Hendricks and will talk to us about SUMICS, Bridging the Diversity Gap in Human Genetic Studies. Adele, are you ready?
Yeah, I'm ready. Can you hear me?
Yes.
Okay, wonderful. Racial and ethnic equality in human genetic research, something many of us have never thought of. But as we see in blue on the left-hand side of the screen, over 75% of these questions have been done on individuals of only European descent. This is a problem because there are so many questions we still need to answer. Like, are there genetic factors that make African-American men twice as likely as white men to develop and to die from prostate cancer? Or are genes influencing the unusually high rates of diabetes we see in Mexican Americans and Puerto Ricans? In order to answer these questions, researchers not only have to gather the genetic data of individuals with a certain disease, they also have to gather genetic data of individuals without a certain disease to use as controls. Let's think about an example. Say a scientist is interested in studying alopecia, an autoimmune disease that causes hair loss. The scientist wants to diversify human genetic research, so they restrict their study to African women. They start by collecting genetic samples from African women experiencing alopecia. Then they have to find their controls, genetic data from African women who are not experiencing alopecia. Now that's a whole lot of data to gather. This project is going to take large financial resources, not to mention quite a bit of time to get up and running. So the scientist speaks with their colleagues and learns of a database called gnomAD that holds previously collected genetic control data. But upon navigating to gnomAD's website, the scientist finds that gnomAD has pooled its African and African American data into one category. This is an issue because historically, African Americans have African ancestry as well as European ancestry. So is there a way that scientists can still use gnomAD's African American data as controls when studying a strictly African population? There is, and it's called SumX. SumX is an algorithm built by Audrey Hendrick's research team at CU Denver. SumX is able to take mixed genetic summary data, like gnomAD's African American data, and estimate how much of that data is strictly African. Then SumX is able to mathematically adjust the mixed data to make it reflect a strictly African control group. So the scientist applies SumX to their mixed data, gathers the strictly African output, and proceeds with their study, knowing they have done what is necessary to ensure the results are reliable and to hopefully make a meaningful contribution to the medical care of an underserved population. While we all work together to bridge the human research diversity gap, there are going to be a number of roadblocks that stand in our way. And while SumX won't solve all of those problems, it will at least make the road significantly cheaper. Thank you.
Thank you very much, Adele. I think another incredibly important topic. Remind us, in which program are you studying?
I am in the Master's in Statistics program at CU Denver.
Yes, and again, for all those who thought statistics is rather dry, I think you have learned aspects that are certainly not dry at all. And how did you come to talk about this topic?
I work at Audrey Hendrick's research lab at CU Denver. I've been working there for about 2 years. And so that's just a portion of what they work on in her lab.
Yes, and I assume that this is mostly computational. So were you still impacted by COVID?
No, it was mostly remote work to begin with. So luckily we could keep things running as normal.
Yes, right. And how much longer do you have to go?
2 semesters.
2 more semesters. Maybe you make another jump forward here. I think it would be very valuable to have even more progress. Do you have any idea where you want to go once you will never be done, but once you have finished your work on your thesis?
I'll probably move into working in the biotech industry.
Yeah, and is there a lot out in this arena? You have an idea if there's a high demand for people with your...
Yeah, I think genetics is a really up and coming field. We've developed quite a few new technologies that make things possible that were never possible before, that we couldn't have dreamt of, just the sequencing abilities that we have. So there are a lot of opportunities for people to do data analysis on the genetic sequenced data.
Yes, and I think there are other disciplines that are then called biostatistics, but actually, while your program is not called this way, it would fall under that. Is that correct?
Yeah, oh yeah, it definitely falls under biostatistics, just specifically genomics within biostatistics.
Yes, yeah. How many people overall in your group are working on these issues?
In the lab, there are about 15 members at any given time. So it's a pretty big sized lab. Everyone has different projects that they work on, but they're all interconnected and we all work under the umbrella of genomics.
Yeah, that's a pretty big group, and I wish you luck with the progress. Hang in there for the next 2 semesters, and all the best. Thank you for being
Thank
here.
you.
Our next presenter is Melissa Schofield. She studied microbiology at the University of Nevada, Las Vegas, under the guidance of Dr. Boshang Sen. And she will talk to us about investigating mutations of a deadly respiratory pathogen. Melissa, are you ready?
I am. Thank you. Today, I'm going to tell you about a pathogen with an interesting mutation, but I first need to talk about cystic fibrosis, more commonly called CF. CF is a genetic disease that causes a buildup of mucus on the lungs and other organs. And as shown at number 1, CF patients suffer from repeated lung infections from a variety of different bacteria. Now, one such bacteria is of great interest in our lab called Pseudomonas aeruginosa. This bacteria forms chronic infections in the lungs of CF patients. And as shown at number 2, these bacteria in the CF lung environment sometimes acquire a mutation that cause them to become thick and slimy. We call these slimy bacteria mucoid. Now, I want you to imagine trying to breathe with slimy mucoid bacteria inside your lungs. It's not hard to imagine how this conversion from normal to slimy or non-mucoid to mucoid results in increased patient mortality. And this is exemplified by the graph at number 3. This graph shows lung function decline in CF patients, where the y-axis is lung function and the x-axis is patient age. The dashed line through the center of the graph shows the point at which these patients will need a lung transplant to survive. You can see patients with non-mucoid infections stay well above that line, but patients with mucoid infections drop below that line, demonstrating how deadly this conversion can be. Now, to make things even more interesting, in that CF lung environment, these mucoid bacteria sometimes acquire a second mutation, thus reverting back to a non-mucoid state. Now, this is thought to happen because this mixed bacterial population may be stronger than either type on its own. However, these non-mucoid bacteria are more susceptible to antibiotics because they've lost the ability to produce that slime and protect themselves. So if we can influence this reversion, we can better treat these infections. The problem is we currently don't know what factors influence this reversion. And this leads me to my current project, shown at number 5. I'm currently investigating different factors that may influence the reversion rate. I'm looking at both host factors and current treatments that CF patients undergo from a very young age. The goal of the project is to see if I can identify a factor that increases this reversion rate. If I'm able to increase this rate of reversion from mucoid to non-mucoid and have it happen in a larger number of bacteria, this population becomes more susceptible to antibiotics. That could lead to better treatments and increased survival for CF patients. Thank you.
Thank you very much, Melissa. Another interesting topic. I'm really fascinated by all you are sharing. Remind us in which program you are.
So I'm in the microbiology sub-concentration as part of the School of Life Sciences at UNLV.
I see. All right. And how did you come to work on that topic?
It's kind of interesting. So when I was looking at graduate schools, it was really my PI's lab that I found interesting. Pseudomonas aeruginosa is an organism that we find in the environment and in water and in infections. And so it's applicable in so many different fields that I thought that was really, really interesting how it can affect, water treatments and plants and of course, human infections. That combined with love for genetics led me to this project specifically.
Yeah, interesting. So you even foresee far-reaching opportunities once you have figured this out.
Yes, definitely.
Yes. And has your work been impacted by COVID?
Yes, so I was very fortunate to be able to stay in the lab even through most of shutdown. And I think, like a previous presenter said, after a while we had shifts. So I really did get to stay in the lab through most of it, which was fortunate that my work didn't fall too far behind. But of course, there were a lot of like mental health aspects and family aspects that are affected by, a huge pandemic and how I can progress my work with all of that going on. So there's definitely effect.
Yeah, definitely. Even if you are lucky and can work in shift, even having a different population of your coworkers there that is shifting all the time. Yes, I can imagine, not to mention all the other things in the background that are invisible.
Right.
How much longer do you have to go?
Hopefully graduating this December or maybe early next year.
Well, you have our best wishes for a speedy and successful completion. Thank you very much for being here. Our next presenter is Shubham Soni. He studies in pediatrics at the University of Alberta under Dr. Jackson Dykes' mentorship, and he will talk to us about ketone therapy as a new approach to infection. Shubham, are you ready?
Yes, I am. And you can hear me, I assume?
Yes.
Awesome. It's the middle of the summer in Paris, and this is it, the final stage of the Tour de France. Your teammate approaches ahead of the pack. You pass off the satchel with the food and drink he needs, and you're confident he'll finish strong, all because of the special legal mixture in the water bottle. Ketones. That's right. Studies in elite cyclists have shown that this unique ketone drink formulation can boost their performance and give them just the edge they need to win races. Some of you have probably heard of ketones in the context of the ketogenic diet, but the inner workings of ketones are far more complicated, and using them to treat diseases is the focus of my research. Ketones are normally known to be an additional source of energy, but they also have non-energetic properties as well. So, for example, we now know that ketones can reduce inflammation in a variety of diseases. But we have yet to test if this is true in one life-threatening condition, and this condition is known as sepsis. Simply put, sepsis is the body's extreme response to an infection that's usually bacterial or viral. Sepsis causes excessive inflammation and results in organ damage and ultimately even death. And the World Health Organization estimates that sepsis accounts for almost 20% of all global causes of death. Sepsis is treated by using antibiotics that directly fight the source of the infection. But this takes time. While an antibiotic may be working to kill the bacteria, this time allows the live bacteria to continue triggering the immune response, resulting in more inflammation. And this inflammation is the key process that allows an infection to cause more injury by spreading to the rest of the body. And this principle is also how COVID-19 works as well. Currently, no treatments exist to prevent the inflammation and organ damage that occurs in sepsis. And thus, I predicted that I may be able to use the anti-inflammatory properties of ketones to treat this. So in a mouse model of sepsis, which has lots of inflammation, I gave mice the same ketone drink that these elite cyclists use to determine if ketones can reduce inflammation. And to my pleasant surprise, I found that it had an exceptional effect, showing a huge reduction of harmful inflammatory proteins, both in the blood and many vital organs as well. With more studies, we may one day be able to use ketones to treat the many diseases with excessive inflammation and help the millions of people and families affected by infectious diseases and sepsis worldwide. Thank you.
Amazing. Thank you so very much. Remind us in which program you're studying?
It is in pediatrics, technically.
And okay, that's where just the program belongs. That's different at different universities. And how did you come to talk about this fascinating topic?
Yeah, so actually, I came into my lab looking at heart research, and back in 2016-17, there was a big upsurge in looking at ketones for heart failure. And since then, these ketone drinks came out, and they're different. They don't work like a diet, but they work as a supplement. So we wanted to test the effects and see if ketones can be anti-inflammatory and to what extent can they be beneficial. And so we decided to venture into the area of sepsis and use models that we can use to test if they're effective against reducing inflammation. So long-winded way of saying, I studied something else, but came to this topic, and it's been a blast.
Yeah, right. It sometimes happens, but I think this is a fascination with science where so many doors open and you just wish you had more hours in the day. Did COVID impact your work?
Yeah, so most of my stuff, all of my stuff is wet lab in the bench. So just like my peers as well, as everyone has said, inability to go in for a good part, but after I think a few months and a few hard lockdowns, we have it harder here in Canada than the States for the most part. After a few different lockdowns, we were able to find schedules. So yeah, go back in, stagger our work, and try and get going as much as we could.
Yeah, just let's hope that this ordeal is over soon for good.
Yeah.
How much longer do you have to go?
Either 4 months or 1 year. I'm not sure yet.
All right. So with that, I thank you very much, Shubham, and we welcome our next presenter. Suk Mun or Alice Wong is a student in computational and data sciences at Chapman University. She studies under the guidance of Dr. Yuri Maus, and she will talk to us about whodunit, explorations of the sense of agency with neurostimulation. Alice, are you ready?
Yes, can you hear me?
Yes.
Great. Hi, everyone. Have you ever felt like a puppet moving without a will? And no, I don't mean existentially. I mean something more concrete, like a muscle spasm, or the weird kind of jerk you get when you're just about to fall asleep. Where does that movement come from? It's certainly your own hand or leg jerking, but it doesn't feel like you doing it. That's what it feels like when you don't feel a sense of agency over your own movements. When you do, that's just normal life. My research looks at how our brains construct the sense of agency, or more formally, the subjective sense of moving and controlling your body to make things happen. But first, why should anyone care to know how brains construct a sense of agency? For one, not everyone has a typically functioning sense of agency. People with schizophrenia, Tourette syndrome, or dissociative identity disorder, formerly known as multiple personality disorder, for example, have a very inconsistent sense of agency. If we know how the sense of agency is supposed to work in typical brains, we might one day be able to alleviate the distress that comes from not having a consistent sense of agency. So, how exactly do I study the sense of agency? It's usually tricky to decouple movement from a sense of agency after all. Muscle spasms don't happen very often. But in the lab, I can do this with electricity. Using electricity, I can stimulate the portion of your brain that controls your thumb and make it twitch involuntarily. This feels like the knee-jerk reflex you have when a doctor taps you just below your knee, except in this case, the stimulator taps your brain and the reflex makes your thumb move. Normally, this is no big deal. I stimulate your brain, your thumb twitches, fun, but not too interesting. You weren't intending to move your thumb anyways, so you have no reason to think that the thumb movement is coming from you. But what if I ask you to voluntarily twitch your thumb at the same time and I don't tell you when the stimulation is coming? Do you think you could tell who or what moved your thumb? Spoiler alert, my results suggest that you can't always tell, especially if the stimulation is coming around the same time as your own intent to move. What could these results mean? Well, if there's a predictable time window where stimulation-induced movement and your own voluntary movement can be confused with each other, it may be possible to make voluntary movements feel involuntary or vice versa, make involuntary movements feel voluntary. Further brain imaging research could then help us understand the difference in how the healthy brain represents voluntary and involuntary movements. In the end, this research could be the foundation upon which neurological and artificial prostheses can be developed to restore a proper sense of agency in those with a disordered sense of agency. My name is Alice Wong, and this is my 3-minute talk. Thank you.
Thank you very much, Alice. This is exciting and frightening at the same time, I think. So thank you very much for sharing this. In which program are you studying?
I study in the computational data sciences program, but I do research at the Brain Institute at Chapman University.
Yeah, right. So this is another example how important it is for different disciplines to work together, right? Thank you so much. I have no problem to see that this is fascinating, but under the many topics that you could have chosen, what drew you to this one?
So my PI has a general program of research in voluntary action, voluntary movement, and I am highly interested in the sense of agency specifically because it's about the perception of voluntary movements. So I find myself a lot more interested in sort of the cognitive aspects of all human perception usually. So when applied to movement specifically, it becomes kind of the sense of agency. And so when he noticed there was this odd illusion phenomenon that could be induced with TMS, the transcranial magnetic stimulation, which is the neurostimulation, I jumped on the project.
Yeah, right. It's really fascinating. Puts a different spin on the whole issue of free will, even, right?
Exactly.
Has this COVID impacted you?
Absolutely. So my research deals with human participants. And so the thing with, even though I was allowed to be in the lab earlier than, let's say, most people, that didn't help much because there were no participants if the students were taking online classes. So yeah, that was quite a difficult time, but luckily things are starting to pick up now. I've collected more data in the past 3 weeks than the past 12 months.
Yeah,
So
somehow, yeah, it will work out. I think you have to catch up, but then you come back in the rhythm. How much longer do you have to go?
I am going to be graduating somewhere between summer and the end of this year.
All right, well, 2022 graduate, best wishes for your research and your future plans.
Thank you.
Thank you. Our next and last presenter is Tori Stevenson. She studies in environmental science at the University of Idaho under the mentorship of Dr. Zachary Kaler, and she will talk to us about intercropping to promote soil health. Tori, are you ready?
I am. Is my sound coming through okay?
Yes.
Okay. From the year 1900 to today, our global population has increased from 1.5 billion to nearly 8 billion people. By 2050, it's projected to near 10 billion, and the lives of every one of those people will rely on the food, fuel, and fibers grown by agricultural producers. Now, already increased demand for agricultural products has resulted in the intensification of agricultural practices that degrade the health of our soil, its ability to recover from disturbance and be productive both now and into the future. Supporting our growing population is going to require identifying diverse growing practices that promote our soils. So for this reason, my research is looking at one such strategy, which could impact soil health in two key ways: nitrogen use and soil microbial diversity. So what we're doing is growing a type of crop called pulses in alternate rows with barley, a practice called intercropping. Now, pulses are special because they can obtain nitrogen from the air, and this is a huge deal. Nitrogen is the key component in chemical fertilizer. And when we fertilize our soils, we can grow more crops per acre, but it's a delicate balancing act. See, for one thing, fertilizer is expensive. And when we add it all at once, it can be difficult for the crops to take up what they need before that nitrogen is lost from the soil, either converted to harmful greenhouse gases or washed into our waterways as pollutants. So adding nitrogen in excess of what our plants can take up is a waste of a resource with harmful environmental consequences. Now here's where the pulse crops come in. Because they can obtain their own nitrogen, they may give us a better chance of having the right amount in the soil at the right time for the plants. And this has the potential to reduce not only our fertilizer need, but our fertilizer waste. And indeed, what we're seeing is that with intercropping, the nitrogen in the soil is used up more completely, meaning there's less left over at the end of the growing season to have those harmful impacts. So this is great news. But what we also need to remember is that soil isn't dirt. It is a living organism. It's full of microbes that do things like cycle nutrients and protect plants from diseases, maintaining the stability of our ecosystems. So when our soils face a disturbance, like an extreme weather event from climate change, not all of those microbes will be affected equally. What this means is that a more diverse microbial community is going to be better able to recover from that disturbance and keep functioning. It's healthier. And in fact, what we're seeing is that intercropping does support a more diverse microbial community than growing barley alone. So this is also great news for soil health. Now, taken together, these results suggest that intercropping could be a very promising strategy for promoting the health of our soils, but also the production of those goods that we rely on. The impacts of this work will be important not only for producers, but also for the people who rely on them for the food they eat every day, which is all of us. Thank you very much.
Thank you, Tori. I think it is really wonderful to see how important issues all of you address. And I think this is a tremendous work. Thanks for sharing it. How did you come to work in this area?
A little bit by accident, honestly. My background is in more of social sciences. I went to Colorado State University and studied human dimensions of natural resources, but realized I really was drawn to doing the research and the natural sciences. So ended up working in a soil lab at CSU and just kind of fell in love with how important this is. You know, we don't think about the soil, the dirt, the earth that we walk on every day, but it is the foundation of, you know, so much of our lives. And just really felt like somewhere I could make a difference and kind of contribute to science. So I fell in love with it.
Yeah, right. And make invisible agents visible by pointing out how important they are. Were you impacted by COVID?
You know, of course we all were, but I'm pretty fortunate that I was not significantly impacted. Fortunately, here in Idaho, we actually live in a pretty small town. Our lab is pretty small and much of our field work is outside, which is phenomenal for social distancing. So I was able to carry on relatively unimpacted, fortunately.
Yeah, okay, well, that is good to know, at least in this area of your work, while you probably couldn't shut it off completely from other aspects of your life. How much longer do you have to go?
Fingers crossed, I will be graduating this summer.
Yay, all right, well, you certainly have our very best wishes. And what are your plans after graduation?
So I just accepted a position to stay here at UI and start a PhD program, working in my same department on a different project, which I'm very excited about.
Okay, well, thank you very much for being here, Tori, and thank you to all of our presenters. I'm happy to share that we have the results, but I really want to have a shout out to all of our presenters. They are the winners of their university, so with internal competitions, and in our case, even 2 campus competitions. And I just think everyone here who talked and did an excellent job could convince you that our graduate students are the workforce of progress. I'm myself always very happy that I'm not a judge, because I find that incredibly difficult, and want to thank our judges again for taking on this difficult task. Making a distinction between excellent and excellence is hard. I'm happy to share that our first winner is Alice Wong, and our second winner is Melissa Schofield. I applaud both of you, and I will see you again, and hopefully you all will be there too, in our final session, where for the final competitions, where you will compete against the winners of the other 2 parallel sessions. I thank you all for being here. I thank you, our presenters. Take up other chances where you have. This is really, really hard to decide. Thank you for our judges, and good luck with your work and with the upcoming graduations. Thank you all for being here. Bye.